And I'm going to present our group's work on phase separation of intrinsically disordered proteins using polymer field theoretic models. We are a theory and computational group at University of Toronto, and our group leader is, or PI is Professor Yusan Chan, and rest of these are my collaborators in the lab. So we all know when a protein chain comes out of the ribosome, it's just a linear chain. But for some proteins, it folds into a 3D structure, and that determines its function. But for some other proteins like caprin-1, say, in the single molecule level, it doesn't fold into a 3D structure. It stays in a random coil-like structure. But in greater numbers, they can form these phase-separated droplets, which gives them functions. And this kind of phase separation is responsible for different biomolecular condensates. Some of them are nucleolase, disgranules, and etc. etc. So our main aim here is to compute the phase diagrams, because the phase separation is important for those IDPs. And for this, we want to actually compute the thermodynamic equilibrium properties. And here I'm showing phase diagrams for a binary system. Here, given a temperature, the system can phase separate into two different densities, but the equilibrium condition is that for all the phases, the chemical potentials and osmotic pressure should be same. And depending on the interaction types, we can have both UCST and LCST in a single phase diagram, or a single type of UCST phase diagram. So we take a little bit different route to compute these kind of phase diagrams. We start from conventional bead spring chains as representations of proteins, where these beads represent the amino acids and springs covalent bonds. First, we write down the interaction Hamiltonian in terms of bead coordinates, but instead of solving the equations of motion for each bead, we introduce auxiliary fields, which are conjugate to fluctuating densities in the particle picture, and then construct a field picture Hamiltonian. And rest of the things how we do it with manipulating this field Hamiltonian. The advantage of doing this is that we can analytically solve some part of the theory. One of the technique is random phase approximation. And here, with this approximation, we can actually compute the free energy density analytically. And this free energy density encodes chain connectivity as well as amino acid sequence. With this analytically available free energy, we can then compute phase diagrams in minutes on a laptop. Of course, we can also solve the whole field theory, but in a lattice simulation. The corresponding technique is called field-theoretic simulation. And here the fields follow this complex Lange-Java evolution equation. And this is a typical trajectory of this field-theoretic simulation technique. In the panel B, the system doesn't phase separate, where these red clouds represent polymer density. But at low temperature, we see at equilibrium conditions a nice droplet. So in this technique, you can also compute average chemical potentials and osmotic pressures, from which you can construct phase diagrams. But more useful things from this kind of simulation we can compute is the pair correlation functions, which are also useful for interpretation of experimental data. One of the first evidence came to show that the sequence, not only the composition of a IDP is important for phase separation, was the experiment done on DDX4 protein by the group of Julie Formanke from our university, actually. So they showed that the wild type variant of DDX4 phase separates in ambient conditions, but a Charles Campbell variant of the same protein, where they just rearrange the positive and negative charges shown in red and blue, that variant doesn't phase separate. So what happens is there in the wild type variant, the charges are blocky. These red and blue are positive and negative charges. But in the charge scramble variant, they are evenly spaced. So this was one of the first evidence to show actually sequence not the net charge that matters for phase separation. Inspired by that, people have constructed synthetic sequences. These are all 50 mers, which have 25 positive charges and 25 negative charges. All are charge neutral, but their block sequence charge patterns are different, block sizes are different. There are two proposed measures to compute the block sizes. One of them is called sequence charge decoration parameter, SCD. And here sigma alpha is the charge of a residue at the alpha position on the sequence. So here you can see this in this parameter measure, the non-local contacts gets extra weight. So this parameter actually prioritizes non-local effects. But there is an alternate measure which is called kappa. In this measure, we scan through the sequences in blocks of 5 or 6 residues and measure the local charge asymmetry. So this measure actually prioritizes sequence local properties. We can compute phase diagrams for all these 16 sequences and determine their critical temperature, upper critical solution temperature, both in field theory simulation and random phase approximation. One of the noticeable things is that here the blocky sequences tends to phase separate more, and both analytical theory and simulation rank ordering wise matches quite well. Next, we wanted to dig deeper and wanted to see how the contact order actually affects different phase separation properties of these kind of sequences. For that, we computed the radius of gyration for these sequences in the isolated chain level at two different temperatures. And when you plot the RGs with SCD, they line up quite well, very well actually, for both the temperature. But for kappa, there is no visible correlation. On the other hand, this is a single chain property, of course. And we compute a multi-chain property like critical temperature. The performance of SCD and kappa both are not so good and not so bad. So from this, we can conclude that the single chain properties are mostly dominated by non-local properties of the sequences. But for multi-chain case, situation is a little bit different. It's a combination of both local and non-local effects. So what happens is there in a condensed phase, if we take a single polymer inside the condensed droplet, it mostly stays in an extended state. So here, because each bead can interact with different chains, it's okay to stay in an extended state. But at the same temperature, if you look at an isolated chain, it stays in a collapsed state because it has only itself to interact with. These kind of things, features also shows up in contact maps. In the lower half, we have isolated chain intra-chain contact maps, both in MD and FTS. Both shows highly peaked contact maps. And on the lower half, we saw the intra-chain contact maps in condensed phase. These are less peaked and more smeared. But another interesting thing is that the single chain contact properties also shows up in the interchain contact numbers. So this basically explains the mild correlation between single chain properties like kappa and SCD with the TCR. So next we know LLPS could be a regulator of biochemical reactions. But here we are showing that the biochemical reactions could also influence the LLPS. For example, we consider this reaction where AB neutral goes to A plus and B minus ions. These are positively and negative charge ion pairs. And in this solution, if we put a bunch of Sb15, this sequence, we see an re-entrant phase separation with increasing salt concentration. But if we don't allow this as ion association, and the reaction is one way, then this re-entrance disappears. But this is not the only thing here, is that the reaction rate or dissociation constant, it also gets affected by phase separation. For example, in this plot, the black curves are for polymer empty system, but the red curves are for when the system is phase separating with polymer present. Another property of LLPS or usefulness is to organize biomolecules in the condensate. One famous example is this frog molecule where F5 and NP1 further subcompartmentalize in a phase separated droplet, or a ternary-like phase separation. So we wanted to know what could be the possible mechanisms behind this further subcompartmentalization. So we studied co-phase separation of these kind of charge sequences. And we found that when two charge sequences are very similar, like Sb25 and Sb28, in a phase separated droplet, they mix. But when we pair Sb1 and Sb28, which have like very different charge sequences, in a phase separated droplet, they demix. So our conclusion is that this kind of amino acid sequence pattern could determine whether two proteins could demix in biocondensates. Another effect of LLPS would be regulating the dielectric environment. Once the system phase separates, we have a high concentrated region of proteins which have very low dielectric constant compared to outside where we have water. So low dielectric constant means high strength of electrostatics, but does this, we ask the question, does this affect LLPS? So it does actually. We can compute, to model this, we considered a dipole solvent where the solvent molecules can have either permanent dipole or polarizable dipole moment. And when there is no dipole moment, non-polar, the solvent is non-polar, the corresponding phase diagram is this black curve. But when you turn on the polarity, the phase separation propensity actually increases. This was the effect of dielectric environment on LLPS. So we tested our theories or electrostatics-based models with an actual experiment. The experimental results came from this, again, Julie Forman-K lab, when lead author Taihun came. The specific system was Caprin-1, and the main aim of this experiment was to show whether unhydrolyzed ATP affects phase separation other than providing energy through hydrolyzation. So it does actually. In the experiment, they saw that at really low ATP magnesium concentration, Caprin-1 doesn't phase separate. At intermediate salt ATP concentration, they phase separate. At really high, the droplets dissolves. But that doesn't happen for the phosphorylated variant of this Caprin-1 protein, where this phosphorylated variant phase separates only at very low ATP magnesium concentration. So one interesting property of Caprin-1 is that it has +13 charge, which almost behaves as an polyelectrolyte. But the pure Caprin variant has a net charge -1, which kind of behaves as a polyampholite. So in our field theory simulation models, we model the ATP magnesium complex as a 6-bit polymer with net charge 2- and Na+ and C- as single bits. We computed the polymer protein-protein correlation functions at different ATP magnesium concentrations. So we see that at intermediate salt ATP concentrations, the proteins are more likely to interact with each other favorably compared to very low and very high ATP magnesium concentrations. But this non-monotonicity doesn't happen for POI Caprin-1. So this basically indicates the similar reentrance as they saw in experiment. And this trend is also shown in our density snapshots from our simulations. Here, the green cloud is protein concentrations, where for Caprine-1, we see this reentrance, but for PY Caprine-1, no reentrance. And consistent with the experiments also, we see when there is a droplet, there is a co-localization of ATP magnesium concentration with the droplet. And this is consistent also with experimental data, which I'm not showing here, but it is also shows up with our correlation function calculations also. So not only that, in experiment, they can actually compute how likely the ATP magnesium complex is to interact with a specific residue of amino acid in the Caprine-1 sequence. And the green squares are the experimental data. And in our simulations, we can compute an equivalent quantity, which is an integrated correlation function. And our results are shown with red circles. So we didn't attempt to match it quantitatively, but the trends are very similar. And they can, in experiment, they can also measure the potential energy on the residue surfaces, which are shown again in the green squares. These are the experimental data. And in our simulations, we can compute potential of mean force. Actually, what we have done is that in the experiments, they introduce positive, negative, and neutral magnetic particles in the system, and they measure the potentials based on their interactions with the actual system. And in our simulations, we did a similar thing. We basically introduced very small amount of positive and negative charges in the original system, and then computed the relative difference between their positive and negative test charges, potential of mean force. And from that, we can compute a potential energy for each residue. Our results are shown again in red and orange and blue for the three different ATP concentrations, and the experimental data is green. Again, the trends are pretty similar, and the numbers are also not that bad either. So this exercise explains that, at least for this Caprine-1 phase separation, electrostatics is enough to explain lots of things. So in summary, I just showed that only with electrostatic we can explain lots of biological situations, and in the field theory setup, we have done some work with non-electrostatics also, but I haven't shown them here yet because of the time constraint. And the take-home message here is that because the biomolecular condensate contains lots of molecules, it could be really challenging to actually simulate them in silico. But our theories can compute those phase diagrams pretty easily. On the other hand, the chain representations in our theories are not very accurate compared to molecular dynamics. So in that sense, we can use these kind of theories as a complementary method in some cases. But for the cases like the Rajas showed Caprine-1, our theories could be almost self-sufficient. So next thing, we all know that these kind of IDPs, physiologically they operate through phase separation with this multivalent or different types of interactions, associative interactions. But when this interaction types goes awry or bad, these droplets could solidify, which then becomes pathological. And so our hope is that if we study the phase separation properties of these sequences well enough, we can come up with some effective drugs. So all our simulations, we do it with our own Python codes, and some of them are available through our lab website. You can scan this code if you want. And thank you.
Thank you, Tanmoy. We've actually finished well before time, so we can take quite a few questions. There's one from Anand, I think. Yes. During the talk, Anand, are you online?
Yeah, I'm here.
Yeah, so please, yeah. Please go ahead.
You want me to ask the question, Tanmay? Good talk. Or do you want me to read it out to you?
Oh, I didn't read it. Can you ask the question, please?
So the sequences, by the way, FTS is a great model, very fast, high throughput. Of course, the specificity is not there, and that is what I'm coming to. So the moment you have hydrophobic residues, you have
Yeah.
polyelectrolyte, you have positive and negative, right? You don't
Yeah.
have apolar residues into the construct.
So these theories are also, we can include those type of interactions. We have done some work on that. I just didn't present them here yet.
How would you do that? How would you incorporate the solvent accessibility or the molecular surface tension term in that?
No, so basically what I do is that we take a molecular dynamics force field,
Uh-huh.
and so we assume that the molecular dynamics force field like m pi pi or m pi pi recharged, those
So,
are like good
like
enough.
force of force. Those are based like hydropathy scale kind of force field.
Not only hydropathy scale, it's a proper like 20 cos 20 matrix,
Uh-huh.
and so it's not just 20 parameters, but it's like the whole matrix is given, and
Yeah, like
so
the Tirumalai Bittencourt kind of map.
exactly, and we can incorporate those kind of interactions in our theories.
Oh, really? You have written a paper on that?
Yeah, yeah, it,
Can
it
you
can.
share? I will put my email ID. Can you email me that paper?
Sure, sure.
Okay, thanks. Very nice talk, actually. Thanks.
Thank you, Anand. Viman has a question.
Hi, Tanmoy.
Hi.
Great talk. So I'm just thinking that right now the model you have presented, you have only electrostatic unit, right? That means,
Yes,
yeah, the same line of what Anand has asked. You are getting some kind of reentrant phase separation also.
yes,
Yes. So, but it is known that that happens at high salt concentration when the electrostatic interactions are not important,
Yeah.
right?
exactly. That's what
Like,
happening here also actually. So because of this chemical reaction, if you increase the salt concentration to really high, most of the dissociated ion pairs would form these neutral pairs.
okay.
For the hydrophobic context.
It's just okay. So you are supplementing something like this in the form of this. Okay, I get.
Exactly.
And can you tell me that how you get the that intermediate ATP you are getting this phase separation, but not at the high and low? What was your explanation there? I used to get it. The Caprine one.
Okay, the explanation here is that so it's Caprine 1 basically behaves as a polyelectrolyte. So for example, if you take a polyelectrolyte, highly charged polyelectrolyte, it doesn't phase separate at really small salt concentration, and at intermediate salt concentration, you can form these ion bridges, uh,
Okay.
and those actually become mixed and enthalpic or the favorable
At
interactions more
at high
strong.
salt concentration, there are screening.
Exactly.
I get. Okay.
But for polyamphylite, which is POI caprin 1, that doesn't happen because it's if you
I
increase the
got
salt
it.
concentration.
Thank you. Thank you.
Thank you, Viman. So I had a question too, Tanmay.
Yes, please.
So is it possible to think of reentrant phase separation as being similar to uh, the effect of a macromolecular crowder that exerts an excluded volume effect like polyethylene glycol, in this sense that at very high salt concentrations, for the salt to be soluble, it engages too much of the solvent, the water, the permanent dipole solvent, and so the water is no longer available for the protein, for the macromolecule, or for the polymer, right? And so, in a sense, could this high salt reentrant phase separation be akin to the macromolecular crowding that is exerted by something that exerts an extrinsic volume effect?
I agree. I agree. So it's basically here, it's because of the crowding effects of this neutral ion complexes.
Thank you. Thank you, Tanmoy. Thank you for finishing ahead of time.